Great. Started the recording. Okay, so good afternoon, everyone. It's my pleasure to introduce our seminar speaker today, Nastassia Patin. Nastassia did her PhD at the Scripps Institution of Oceanography on chemical ecology of marine sediment bacteria. She then did her first postdoc with Frank Stewart and Kostas Konstantinidis at Georgia Tech in Atlanta, where she applied bioinformatic tools to marine microbiomes of underexplored environments. In her current position, she works with Kelly Goodwin at the Atlantic Oceanographic and Meteorological Laboratory on marine microbiomes and environmental DNA of the California current to understand linkages between oceanographic conditions, microbial communities, and fisheries. In her free time, she enjoys surfing, riding her bicycle, and hanging out with her 2 cats. So welcome, Nastassia. Take it away.
Thank you so much, Jasmine. Can you hear me okay?
I can hear.
Okay, great. So yeah, thank you for that nice introduction. And today I'm going to be talking about my time spent aboard a research vessel called Tara in Antarctica. I was there for a month earlier this year, and I'm really excited to talk about this very and hopefully the NOAA community will find it of interest as well. So this is a photo of me up in one of the crow's nests of one of the 2 masts, Tara, which are about 2 meters high or 100 meters high, 100 feet high. And I had this opportunity to climb up and survey the seascape around me during a day of nice weather when we had finished our sampling on time. So as you can see, the view was really stunning. So as I said, this expedition took place in January and February earlier this year. And I was one of 14 people aboard Tara for this mission. And we were in the Southern Ocean and Weddell Sea, taking part in a much larger project called Mission Microbiomes, which is itself one component of a larger EU-led project that I'll talk a little bit more about in a second. And we were lucky to have a wonderful media person, Maiva, who took this stunning photograph using drones during our cruise. So to give a little overview of how this all came together, NOAA is a partner in a project called Atlantico, which is a multifaceted EU, European Union-led project, a horizon.
I'm sorry to interrupt, Nastassia. Can you all mute your microphones?
Institutions and participants, including the EU, but also participants in Brazil and South Africa. And the goal is to the resources that help the marine community thrive. And so this infographic here shows some of the main components of the research that they are thinking about. So they are targeting 5 ecosystem services, including food security, climate impacts, biodiversity, pollutants, and deep sea life. And then within those, they have these 3 research pillars, which include microbiomes, which is my field of specialty. So I'm a microbiologist turned computational biologist studying microbial communities in the ocean. They're also very concerned about plastics and the effect of plastics in the marine environment. And then this bigger concept called seascape and ocean connectivity, which is more the physical oceanography side. And so they have a number of different smaller projects and cruises that are associated with Atlantico. But its flagship mission is called Mission Microbiomes, and it takes place, is still taking place aboard this boat called Tara. And so Tara is quite well known in Europe, but I wanted to give a kind of an overview of this ship for those of you that may not be familiar with it, because it really has quite a fascinating history. So it's a 36-meter schooner with 2 masts, as I mentioned. There are 14 people on it generally, built for 12 crew. We had 6 scientists and 8 crew. And it's already been through quite a lot, quite a different range, quite a range of environmental conditions and locations. I'll talk about more in a second. But it has a really interesting origin story. So it was built 30 years ago, more than 30 years ago, in France, and it was designed to mimic a boat called the Fram, which was the first boat to complete something called an Arctic drift, which is where it deliberately sails into the polar ice field. And then as the ice closes in, rather than being crushed, the boat is lifted up onto the surface of the ice and remains there over the winter as the ice rotates around the polar cap. And then in the spring, when the ice starts to melt, the boat is released on the other side of the world. And so Fridtjof Nansen's famous expedition on the Fram finally put an end to this theory that was very common at the time. This is the turn of the 20th century, that there was in fact a warm tropical ocean around the North Pole. That if you just got through the ring of ice around the North Pole, you would enter this open polar sea. But his this Arctic drift really put the nail in the coffin of that theory. And so it's quite a famous expedition. And Tara was built to mimic this design and also to complete an Arctic drift. And so on the bottom here, this is a photo of Tara being constructed, and you can see the hull is really strongly reinforced with aluminum so that it will not be crushed. And it also has more of a curved shape on the bottom so that again, it would be lifted up onto the ice. So it never fulfilled this vision the first 10 or so years of its life. And I think its original owners kind of had some financial and logistical issues. But in 1999, Tara was bought by a famous yachtsman called Sir Peter Blake, who had won the America's Cup multiple times, was very famous in the yachting world. And he renamed the boat Seamaster and used it for his company called Blake Expeditions, where he wanted to travel the world and promote environmental responsibility and the effects of climate change. But unfortunately, not long into his mission, Sir Blake was killed by river pirates in the Amazon. And the boat sort of languished for a couple years before it was bought by the Tara Ocean Foundation in 2003, renamed Tara, and became a vessel for oceanographic exploration, where they partner with scientific institutions, academic institutions to do oceanography. And its first mission was Tara Arctic, where it finally fulfilled this Arctic drift vision, which is pretty cool. From 2009 to 2013, it conducted Tara Oceans, which is the most famous of the Tara expeditions thus far, and by far the most scientifically productive. So I'll talk a little bit more about Tara Oceans in a second. There were a few other expeditions between Tara Oceans and Mission Microbiomes, but in 2020, Mission Microbiomes became the flagship expeditions for Atlantico. And so just to show you this comparison between the Fram expedition and Tara Arctic, you can see that in very similar ways, the boats entered the ice fields, were trapped for a period of several months, rotated around the North Pole, and were released on the other side. I think Tara Arctic, it was actually more than a year and a half that they were in the ice. So it wasn't just like a seasonal thing. It was quite a long time. Same for the Fram. Okay, but Tara Oceans is where I first learned about Tara, and any microbiologists in the audience are probably familiar with them as well, because they were a long expedition that conducted this standardized sampling of microbiomes and other oceanographic variables all over the globe. And they, at every station, they filtered massive amounts of water in exactly the same way, preserved the samples, and ultimately sequenced all of these data and made them publicly available. And these publicly available data have spawned numerous publications, hundreds of publications, and it's been a real boon to the scientific community, particularly for marine microbiologists, for anyone interested in phytoplankton, global distributions, global patterns. it's really been just a remarkable scientific achievement. And so I had been using and looking at Tara Oceans data for a long time and not really understanding the context. And these are just a few of the bigger publications that have come out of Tara Oceans, but there are so many that I could list, and they've really been, the data have really been a game changer for those of us that are maybe not able to get out in the ocean where we want to, or are just interested in these big picture global patterns. So it's been a really amazing run for Tara thus far. And in Mission Microbiomes, this is in some ways some of their most ambitious, one of the most ambitious expeditions yet, because the sampling protocols have really been expanded. So in Tara Oceans, they did their omics filtering, which means they're filtering 2 size fractions, 20 liters of water for each sample, as well as sampling for viruses. And that was done in Tara Oceans at every station. But for Mission Microbiomes, they've added many additional protocols. So including what they call eDNA, which is just a different size fraction targeting eukaryotic organisms. They're also sampling for aerosol microbiomes, running samples for HPLC to look at pigments, doing plankton toes, doing biogeochemical sampling, sampling for microscopy. And most recently, they've added a trace metal clean rosette to look at trace metal concentrations. And so this is the new standardized protocol for every station that you see in the Mission Microbiomes map here on the right-hand side. And so far, they've completed, I believe, 13 legs of Mission Microbiomes. They're now somewhere up here near Europe, having just gone up the west coast of Africa. And my leg 11, was down here in the Southern Ocean and Weddell Sea. And this map is not entirely accurate, as I'll show you in a second. This was the sort of predicted or envisioned itinerary, but it's changed a little bit since then. But it's really a remarkable effort that is covering, covering a lot of the important variables in oceanographic research, and it's really amazing to see what this pretty small and scrappy boat can do. So at this point, I want to introduce the team that I worked with on Mission Microbiomes. Like I said, there were 14 of us, including 8 crew, really 6 main crew, and then our 2 sort of added on, our journalists and media person. But these are all crew members that have been with Tara for a long time. They rotate in and out over as the missions progress. They're not all on board for 2 full years, but they have all been with Tara for a while and are extremely competent sailors. They also all have a real interest in the science and in environmentalism. They're really, proud to be on Tara and motivated to be part of such a unique element. And so, that combined with their sailing prowess really made it a valuable team. And then our scientific team was led by Chief Scientist Alessandro Tagliabue, who is up here in the middle, sitting cross-legged here. And Al is from the University of Liverpool. He's a biogeochemical modeler. And so he was very interested in the Southern Ocean for biogeochemistry reasons that I'll talk a little more about later. We also had Professor Chris Bowler from the CNRS in Paris, who's a real leader in the field of diatom biology. So Chris is up here on the right-hand side. And then Léa Olivier, Doug Coué, Thomas Lankowski. Doug and Thomas were both engineers, sort of what we would call maybe technicians, but very knowledgeable, real biologists in addition to their technical expertise. And then myself and our 2 media people. And the crew especially is all French. And depending on the leg, the scientific team can be very francophone or not. In this case, we had everyone on the team spoke fluent French. My French was by far the weakest of the team, but I was able to get by. But everyone also spoke fluent English, and the science was mostly conducted in English, so it was okay I never felt sort of lost or inhibited by that. So Mission Microbiomes, we had 2 main goals in the Southern Ocean and the Weddell Sea. Our first goal was more of a kind of a micro scale, if you will, an iceberg process study. And then our second goal was on a larger scale, assessing an iron limitation gradient across the shelf. And that was focusing on this transect here out from the tip of the South Shetland Islands, or the tip of the Antarctic Peninsula into the Weddell Sea. And these goals were sort of set forth by the chief scientist. So for every leg of Mission Microbiomes, there are these overall goals of the standardized sampling, the collection of massive amounts of data at every single station. But within that, there can be different areas of focus and different specific targets. And so Al set these studies largely in accordance with his research interests, as well as some bigger picture ideas about climate in the Southern Ocean. So I'll just talk a little bit about each of those goals and how we went about achieving them. The first goal being this iceberg process study, where Al worked with the British Antarctic Survey over a number of months, even before we got on board Tara, to identify a large iceberg that would be a good subject of our study. And then he used the satellite data that was transmitted in real time to him as we were aboard Tara to try and track down the iceberg of interest. And that in itself was actually really challenging. Our internet connectivity was very poor, and he wasn't always able to get communication in a timely way. And it turns out these icebergs move around quite a lot. And so by the time we sort of thought we knew where one was, it would have moved. But eventually we found this quite large, I think it was about 5 kilometers squared in area, and quite tall, as you can see from this photo. And we identified this target, and we kind of went with it. And so the scientific goals here are really around some of these microscale processes that happen around an iceberg. So icebergs originate on land, so they're not sea ice, they're really land ice, and they bring with them the nutrients and features of the land that then sort of set adrift in the ocean. And as they melt, they're releasing these nutrients into the seawater around them and really affecting the ocean environment. This is sort of a conceptual diagram from a review paper a few years ago about the many different ways icebergs might affect the surrounding water column. And so we wanted to do this study because there's been so little research on iceberg kind of dynamics and how it affects oceanography. And because Tara is such a unique vessel where we had the ability to do this, we could get really close to these icebergs, we could maneuver around them. And so it was really kind of an ideal platform to do this kind of study. And so for this study, we had 3 sampling designs. This, I should say, this top right photo is not the same iceberg. It's just a photo I used for convenience. But the bottom 2 are our target iceberg. And so we had one day where we did a horizontal transect away from the iceberg, sampling just at the surface, but at 4 different points, moving away horizontally. We had what we called around the clock, which was also surface-only samples at 5 different sites around the iceberg. And then we had a vertical profile where we picked one spot and just did 4 different depths with our CTD cast, and just spent one day sampling that water column vertical profile. And again, in each of these sampling stations or sites, CTD cast, we would perform the exact same suite of protocols so that the data were collected in a really standardized way. And one of the things that Al was really interested in was this trace metal component. And so in addition to our trace metal clean rosette, he's here with a PhD student, Leah, trying to sample surface water in a trace metal clean way using a bow pole. And as you can see, this was a site that was very, very close to the iceberg. And again, on Tara, we were able to maneuver very well and get really close to these structures. And this map on the right here is just showing how far we drifted over the course of this study, which lasted about three full days. And we started in one spot, but as you can see, over the course of these days, the iceberg actually drifted around 30 nautical miles, and we were constantly having to maneuver to stay in place and track it. And it was really, quite a challenge, I think, for the captain and crew, but it was a successful one. We accomplished all 3 of the sampling designs that we targeted. The second goal was more of a bigger picture oceanographic goal, where we wanted to assess an iron limitation gradient in the Weddell Sea. And so, as many of you probably know, the Southern Ocean, unlike other oceans, is highly iron limited. Rather than being limited by nitrogen or phosphorus, it's limited by iron. And so it's what's called a high nutrient, low chlorophyll region, where concentrations of nitrate and phosphate are actually quite high. And DIC, which is shown here on this map, is quite high as well, what they call unused DIC, because there's not enough iron for phytoplankton and other organisms to make use of the other nutrients. And so this is a really important feature of the Southern Ocean, which also has direct effects to all other ocean basins. This is a Spillhouse projection of the Southern Ocean and how it connects to the rest of the world's oceans. And what happens is, in the circumpolar current, this water sinks, becomes part of the deep bottom water, which then cycles around the other oceans and can remain in the deep part of the water column for hundreds of years, if not longer, before finally resurfacing. And so what happens in the Southern Ocean really has important implications for the rest of the chemistry and biology of other ocean basins. So we were interested in assessing this iron limitation gradient that has been studied before in a limited way. And the other component of this is that the Weddell Sea, as much of Antarctica has been experiencing, is seeing lower and lower levels of sea ice every year. And 2022 was actually a record low year for sea ice extent in the Weddell Sea. And so we got down to around the 65 degrees south. So this is again a planned map of our journey. This is not where we actually ended up, but so we got a little farther than expected here for the iceberg study. But we conducted this transect across this iron gradient across the shelf, and this transect has been sampled before by, for one, the German icebreaker, the Polarstern, has conducted trace metal experiments along this gradient as well. And so it was a chance for Tara to show that using the trace metal clean protocols we have on board, they could replicate the results that have been already observed by other oceanographic vessels and sort of prove the metal of the GEOTRACES style sampling that was planned. And if the data are good, then of course they can inform, what was happening in the Weddell Sea at that time of year under those circumstances, and combined with all our omics data and other biogeochemistry data, we can start to put together a really amazing picture of the ecosystem along this iron gradient. So I want to talk a little bit more about the work aboard Tara, because it was really so unique and so different from any other research field experience I've had. There was, as I've mentioned, the standardized sampling workflow that we were meant to follow at every sampling site, and the protocols were divided among 5 operators. And then we had 6 scientists, which not every team has. It depends on the logistics of the team. But we had a 6th scientist who, in this case, was our chief scientist, Al, who could sort of float among the different protocols and help out where he was most needed. And what happens at each station is CTD cast is done, and the rosette brings up a good amount of water that gets divided among these large filtrations, which include omics, which always use at least 60 liters of water, because there are triplicate sampling filtrations of 20 liters each, as well as a smaller one for what they call eDNA. They differentiate their eDNA from their omics protocol. And then there's also a series of smaller filtrations, which I was in charge of. That was my operator role. Although my expertise is really more on the omics side, I handled these small bottle filtrations to preserve samples for microscopy, microplastics, HPLC, and various other protocols back on land. We also at each site tried to deploy this trace metal clean rosette. If any of you have done trace metal work before, you know that this is extremely delicate and involved work, and so we were not able to do it at every single site. But Tara, unlike a lot of big oceanographic vessels, Tara is not made of iron or steel, and so in some ways that made the job easier of keeping it trace model clean, but in many other ways, especially the small area that we had to work in, and a lot of the other constraints that are on a small vessel, it was a really, really challenging part of the protocol. So I'll be very interested to see how those data look down the line. We also, part of the standardized sampling was also plankton tows, looking at zooplankton and phytoplankton in each of these sites. And so I'm shown here on the left, collecting one of my smaller samples for one of these smaller filtrations. And being Antarctica, you know, most days were quite cold, but we had pretty good luck with the weather overall, I would say. Especially, you know, we were able to complete this transect going out into the middle of the Weddell Sea Gyre, which was, you know, very subject to bad weather. But you know, even under snowy, windy conditions, we were able to get the CTD in the water and get these types of samples. On the right-hand side is what they call the wet lab aboard Tara. It's a very, very small space, but it's very well organized, well set up. You see these carboys here that each contain 20 liters of water and are used for the omics filtrations. And that water is piped directly into the filtration setup inside the wet lab. And it's really, they have a very streamlined protocol to run these filtrations. And it's really remarkable how much water you can filter when you have this efficient and streamlined setup. So this is the inside of the wet lab. You can see a series of peristaltic pumps on the right-hand side leading to these large 142 millimeter filtration setups. It's all very well well set up, and the space is very well utilized. You have some smaller filtration setups over here, and some of the biogeochemical sampling that is done in that space as well. And then not shown over on the left side would be the chemical fume hood and the setup for aerosol sampling as well. And then down below, we had what they call the S lab, which is where the small scale filtrations happen. Each sample was split into equal volume little mini samples seen in this little brown bottles here. And so this was a sample that was particularly high in diatom, levels of diatoms, and you saw the water was really very brown as a result of that. And Tara also has this instrument known as the flow cam, which can image phytoplankton at a single cell level, which was really, really cool. And Chris Bowler, our resident diatom expert, managed the flow cam and really produced some beautiful images of phytoplankton in real time. And as a microbiologist, it was really cool for me to see this happen. You know, I never get to really see the things that I'm studying because I'm always just filtering the water and saving filters. But here we could really take a look at the phytoplankton community and see some beautiful shapes and designs of all the slightly larger microbes that are in the water column. So another aspect of life on board Tara that I wanted to talk about was the non-work aspect. This was a really highly communal and collaborative work environment. Everyone, because we were such a small team, everyone really helped out with everything. And so that included chores on the ship. We were all assigned to be on a team. The teams always included at least one scientist and one crew member. And every day we rotated through certain chores. And so on any given day, a PhD student might be cleaning toilets with the captain of the ship, or a technician might be washing dishes with the chief scientist. And this kind of really interactive environment was, I just thought, so remarkable and really made for the opportunity to get to know everyone on the team really well and kind of treat everyone as equal in their roles and responsibilities, because truly everyone had a critical part to play aboard Tara, and we all, I think, felt that sense of responsibility as well as privilege. You know, everyone sort of felt very deeply that this opportunity was incredibly unique, and as a result, we have this amazing responsibility to do the best work, the best science that we possibly can, and as well to communicate the importance of that science, you know, as well as possible. There was also, as I touched on before, this very strong francophone culture. So all the crew members are French. The Tara Ocean Foundation is a French nonprofit foundation. And so the culture around meal times, around free time was very French. And I personally really enjoyed it. My French is not as fluent as it could be, but I was able to get by really well and enjoyed my conversations and enjoyed the chance to have this sort of multicultural experience. And so, yeah, for me, being aboard Tara was really a once in a lifetime field experience. The other component that I've talked a little bit about is this logistical flexibility that we had aboard Tara, because it was so small and maneuverable, and because we were not tied to some large, institutional schedule. We were able to do these things like the iceberg process study. We were also able to go on land quite a few times as sort of the schedule permitted. And so here are just a couple more photos of sort of life aboard Tara. On the left was our galley, and that happened to be a kind of wine and cheese night. So we had some nice cheeses that were out. Must have been particularly celebratory. Maybe there was a successful sampling day. And then on the right-hand side is where we, as scientists, were enlisted to help with the sailing of the vessel, which, it was really just doing exactly as we were told. There was no sailing expertise was needed to be on board, but it was really cool to see this schooner in action and really take part in the running of the hoisting of the sails on this day. Yeah, so just another example of how everyone kind of did everything. I was enlisted to help fix nets at one point. I remember seeing the cook multiple times, help deploy the CTD. And so we were really, all in on the scientific and the running of the boat together. And on the right-hand side, this is just to show how close we really were able to get, sometimes a little closer than I think was intended. There was at least one instance where we had to sort of back out of this area where the ice flows were a little closer than anticipated. But luckily, again, the crew was just so competent that it was I never felt like we were in danger. And like I said, we had the chance to go ashore several times and see some penguins, see some charismatic megafauna on land. And so that was really, really just an incredible experience as well. Penguins are just truly the most adorable of creatures. And we visited a few sites that were actual big colonies. So this is a map of some of the islands where we were able to go ashore. And we even, in one instance, managed to get on the actual mainland, the Antarctic Peninsula itself, here at this site called Esperanza, where there's a Chilean base. And that was really, really remarkable as well. And zooming in on this a little bit, because of this schedule flexibility, we were able to complete a couple impromptu goals. And so this first one was sampling at hydrothermal vents at Deception Island. So Deception Island, you see over here, it's a horseshoe-shaped island, and it's horseshoe-shaped because it's actually a caldera. It's a volcanic caldera, and there is quite active geothermal activity that still goes on at Deception Island. And there are hot springs that are bubbling up around the inside of this, the inside of the island. And you can actually see, this is a photo of one of the inner beaches, and it's quite dramatic looking, very black sand. And another notable thing is you'll see no penguins and no large animals, unlike really all the other islands that we went to. It's pretty devoid of large animal life, and this is likely just because of the geothermal effects of the bay and the sand properties itself as well. We got to go ashore at Deception Island as well, but for me, this was such a unique opportunity because Al, our main chief scientist, Al, actually handed the reins of power over to me, and I was able to act as chief scientist for a day and direct sampling operations at the hydrothermal vents because, this is a case where the prokaryotic community, the archaea and bacteria, are really going to be the main point of focus. And so Al, sort of recognizing my role in microbial ecology, sort of let me direct the sampling operations. And that was really just an amazing opportunity that I am so grateful for. we did get to go ashore and do a little hike and managed to get a photo of Tara in the bay from the top of one of these hills. It was really, really spectacular. And our second impromptu goal was a hunt for something that was so-called ancient diatom, which had been discovered from a sample in the Antarctic Sound several years previously. This is a diatom with an extremely large genome. So here's some of the stats on the genome size of this organism. And Chris Bowler, our diatom biologist, remembered, the location of this sampling, and we spent a day just exploring the Antarctic Sound looking for this diatom, and he found it. He found it using the flow cam, and we did some sampling at that site, and so we're hoping to do some population genomics on diatoms, this Odontella ancient diatom from the Antarctic Sound. So this again, which just kind of happened because we had completed our main sampling objectives, we had some extra time, and we're able to leverage that time and really maximize our scientific work in Southern Ocean. Tara is also a boat that, especially in Europe, is really amenable to publicity, to media outreach. And while we were in Antarctica, our chief scientist sort of did this live broadcast into a summit taking place in Europe, the One Ocean Summit. And we have this nice image of him all bundled up, trying to speak clearly into the microphone despite the wind, and French President Emmanuel Macron on the right-hand side, looking very, very serious, very concerned with what Al is saying about the Southern Ocean. So that was kind of a cool little experience that we had on the media side of things. We also had an article, our resident journalist, Vincent Joly, published this sort of feature piece about our expedition in Le Figaro magazine, which came out about a month or so after we returned. But these are all very nice and a really important part of what Tara does. But it's really the science that comes out of it that I think is the most remarkable aspect of Tara. And just this month, they made it again onto the cover of Nature Microbiology, and that was because of a perspective piece that they published on ocean microbiome research, coming out of what is at this point more than 10 years of sampling and sequencing and collaboration across many different scientific institutions, and what we've learned so far, about the role of microbes in the ocean, their importance in nutrient cycling and ecosystem services, and the importance of studying them in the context of climate change, and how our changing ocean is really going to be driven by the changes in microbiomes. And so, I think this really encapsulates just the importance of this kind of sampling, the persistence of this sampling, the standardization, and it was really just amazing to be part of that just for a short time. So what's next for the Tara samples? The omics samples will be sequenced in Europe and preliminarily processed as well. And then for the leg that I took part in, I'll be able to have a hand in the sequence data processing and addressing some of the oceanographic questions that we have regarding the Southern Ocean and the role of climate, microbiomes in ocean climate. And other samples will go out to their individual labs, which are specialized in whatever particular topic that is special about those samples. And ultimately, the goal is to come together and really address, tackle some of these questions from a holistic standpoint, and combine the chemistry, the biology, and the physics that we have in these massive data sets. So I'm really, really excited about the next steps here, and I'm looking forward to working with my team members in the future as well. So I will end there, and I'm happy to take any questions about any aspect of this talk. I know it was a lot, and that kind of brushed over a lot of the science, and so I'm happy to talk more about that aspect, or really anything else you might have to say. But thanks so much for your attention.
Nastassia, if people would like to put questions in the chat. or you can come off mute and just ask the question directly. So there's a question, was anyone taking weather measurements?
Weather measurements. Well, let's see. We were sampling for aerosols, as I mentioned, aerosol microbiomes. I don't know specifically, other than sort of, the basic ocean characteristic, temperature, salinity, and biogeochemical parameters. I'm not sure. And I guess recording, of course, the ship crew was certainly recording weather conditions every day. So sea state and cloud cover and things like this were being recorded. Anything more specific, I'm not sure about that. Did that answer your question, or was there something in specific you were wondering about?
No, that was fine. I just a great opportunity to get in some sort of weather measurements. So maybe that's all they took.
Yeah, I mean, the focus is really on ocean sampling, not so much on weather. So probably the basics. Oh, great. Oh, I'm so great to see one of my teammates is here. Leah, who was just an incredible part of the team. Leah says there's a weather station on board with air temperature and winds. And so that would have all been recorded and probably available with all the Tara Oceans data, which is publicly available. There was a question of how much time were we only under sail power? So not a lot of the time, mostly due to the danger of being in a ice flow dominated ocean. So we really had to be able to maneuver very quickly and precisely around icebergs and ice flows. And so not very often, but occasionally when the weather was good and the seas were clear, we did put up sails. And then also on our return journey from the Antarctic Peninsula back across the Drake Passage back to Chile, we were entirely under sail for the 3 days of that voyage. It was apparently a more efficient way of traveling under those particular winds and circumstances. So that was really nice to have no motor and just be under sail for crossing the Drake Passage.
There's another question, Nastassia, I'm not sure if you're seeing it. What a great talk. Any ability on board for DNA sequencing like with Minion? Really exciting.
It's something that is, I think, of course, being talked about. At this point, the protocols are really, really involved, and it would be very difficult to add anything new, especially something as involved as mid-ion sequencing. I think it's definitely a possibility down the line, but at this point, because the omics sampling is so robust and has kind of proven its worth, I think it's not a priority, but I think it's always on everyone's mind these days. Anyone who's works with eDNA or omics and is out at sea, it would always be so amazing to have real-time sequence data. So I'm sure people are thinking about it, but I think for the time being, it's not particularly realistic.
Okay, I'm not seeing anything else in the chat. That was really amazing. Nastassia, thank you so much for sharing your experience. If there's no other questions, we'll end the seminar. Thank you all so much for attending.
Thank you, everyone. I appreciate your attention.
Thanks so much, Nastassia, for the talk.
Yeah. Oh, thank you so much for coming. This is so nice.
Yeah, it was great to see you. It was a great talk. You talk about so much stuff.
I know it was a little long, but hopefully no one minded.
It was great. I have a question for you. It's a small one. Do you plan on going back on tower?
You know, I would definitely consider it. I think they're almost done with mission microbiomes, right? So after that, I don't really know what's happening, but yeah, I'm available. I put my name on a list at some point with Duke, so he knows he knows he can contact me.
Yes, even for mission microbiome, I think they still have a few legs there in Angola, I think at the moment, in Congo.
Oh, really? Okay, I thought they were farther along.
Still some work until Lisbon, but yes, I think they were in the Congo at the moment. I received a message from Doug yesterday.
Although this fall might be a little tricky. Yeah, it's just, it's hard to schedule those long expeditions, but
sure,
it's a possibility. How are you doing? I guess we have it to ourselves right now, which is nice